Very good evening. PUC ಸೆಕೆಂಡ್ ಇಯರ್ ಬಯಾಲಜಿ ಪಾಸಿಂಗ್ ಪ್ಯಾಕೇಜನ್ನು ಡಿಸ್ಕಸ್ ಮಾಡ್ತಾ ಇದ್ದೀವಿ. ಹಾಗಾದರೆ ಇವತ್ತು ಚಾಪ್ಟರ್ ನಂಬರ್ 11 ಬಯೋಟೆಕ್ನಾಲಜಿ ಪ್ರಿನ್ಸಿಪಲ್ಸ್ ಆ್ಯಂಡ್ ಪ್ರೊಸೆಸಸ್ ಅನ್ನುವಂಥ ಒಂದು ಚಾಪ್ಟರ್ ಬಗ್ಗೆ ಅದರಲ್ಲಿರುವಂಥ ಇಂಪಾರ್ಟೆಂಟ್ 1 ಮಾರ್ಕ್ ಕ್ವಶನ್ಸ್ 2 ಮಾರ್ಕ್ಸ್ ಕ್ವಶನ್ಸನ್ನು ಏನು ಮಾಡ್ತಾ ಹೋಗೋಣ ಅಂದರೆ ಒಂಥರ ಆಡಿಯೋ ನೋಟ್ಸನ್ನು ಕೇಳ್ತಾ ಹೋಗೋಣ. ಹಾಗಾದರೆ ಮೆನ್ಷನ್ ದ ಇಂಪಾರ್ಟೆನ್ಸ್ ಆಫ್ ಸೆಲೆಕ್ಟೇಬಲ್ ಮಾರ್ಕರ್ಸ್ ಇನ್ ಪ್ಲಾಸ್ಮಿಡ್ಸ್. Okay, mention the importance of selectable markers in plasmids. It helps in identifying and eliminating non-transformants and selectively permitting the growth of the transformants. ಹೌದಾ, ಇದೇನಪ್ಪ ಅಂದರೆ ಇಂಪಾರ್ಟೆನ್ಸ್ ಆಫ್ ಸೆಲೆಕ್ಟೇಬಲ್ ಮಾರ್ಕರ್ಸ್ ಇನ್ ಪ್ಲಾಸ್ಮಿಡ್ಸ್. ವಾಟ್ ಈಸ್ ಅ ಮೈಕ್ರೋ ಇಂಜೆಕ್ಷನ್? It is the method of introduction of the rDNA, recombinant DNA or recombinant DNA directly into nucleus of a cell, animal cell or host cell. It is called as a microinjection. Name the stain used to visualize DNA fragments in gel electrophoresis method. Okay, it is a what? Ethidium bromide. It is a stain used, which one? Ethidium bromide. Name the molecular scissors used in recombinant DNA or rDNA technology. Restriction endonuclease or REN. Those are called as a REN, restriction endonuclease or restriction nuclease enzyme. It is also called as a restriction nuclease enzyme. Define polymerase chain reaction. Polymerase chain reaction is a reaction in which multiple copies of a gene or DNA is synthesized in vitro using 2 sets of primers and the enzyme DNA polymerase. Mention the technique used to separate DNA fragments in recombinant DNA technology, gel electrophoresis method. What are plasmids? Plasmids are extra nuclear circular double-stranded DNA present in the cytoplasm of bacteria. What is a downstream processing? A series of a process before it is ready for marketing as a finished product. It is called as a downstream processing. Write the restriction site of a E. coli enzyme. E. coli. It's a RI enzyme. Here DNA 2 strands are there. Okay, one strand is 5:3, another strand is 3:5, and whatever coding is there here, GATTC is there. Okay, wherever guanine is there, here cytosine will come. Wherever here adenine is there, in that case thymines will come, AATT. Then wherever here thymine is there, here adenine will come, and here cytosine, here guanine will come. You already studied about pairing up purines and pyrimidines. Based on that, these 2 strands are formed. Retroviruses are disarmed before using to deliver desirable genes into animal cell. Why? To make them non-pathogenic. Retroviruses are disarmed before using to deliver desirable gene into animal cell. Why? To make them non-pathogenic. What is a elution? The separated band of a DNA are cut out from agarose gel and extracted from gel piece is elution. Name the plasmid present in Agrobacterium tumefaciens. Okay, it is a TI plasmid or a tumor inducing plasmid. TI plasmid or tumor inducing plasmid. Okay, 2 mark questions. What is the significance of selectable marker in cloning vector? It helps in identifying and eliminating non-transformants and selectively permitting the growth of the transformants. What are polydromic nucleotide sequences? Write the restriction site for E. coli enzyme. Okay, the polydrome in DNA is a sequence of a base pairs that read the same on the 2 strands in 5:3 direction and 3:5 direction. Restriction site, okay, I already discussed with you. These are 2 strands. Okay, 5:3 one strand, 3:5. Okay, because same codes are not formed in one strand. That's why wherever 5 is there, here 3, here 5, and guanine, cytosine, thymine, adenine, thymine, okay, and cytosine, guanine. Okay, alternatively the pair. Write any four tools used in recombinant DNA technology. Restriction enzymes, polymerase enzymes, ligases, vectors, and host organisms. Draw a label diagram of a stirred tank bioreactor. Stirred tank bioreactor diagram, it's a stirred tank bioreactor diagram, it's a okay. You see this diagram here, whatever the part is there, acid base for a pH control, here it is there. Okay, then whatever this device is, the steam for a sterilization, then motor, foam breaker, flat bladed impeller, then a cultural broth, then a sterile air. Okay, we process from bottom. Then uh, gas entertainment, increased surface area for oxygen transfer, where here then whatever the bubbles are dramatically increase the oxygen transfer area, okay, where inside the tank. Okay, this is the diagram of stirred tank bioreactor. Classify restriction enzymes, mention the function of each. Endonucleases, the cut at a specific position within the DNA, it is called as a endonucleases. Then exonucleases, they remove nucleotides from the ends of the DNA. Draw a neat label diagram of typical agarose gel electrophorese. Agarose gel electrophorese. Okay, these are the main structures. Whenever they go undergo gel electrophoresis method, they form sub bands. Okay, in upper region largest bands are formed, and the lower region smallest bands are formed. These are structures shown here. Okay, this is the neat label diagram of agarose gel electrophoresis. Mention two techniques to make a host competent in RDN, recombinant DNA technology. Techniques to make a host competent in recombinant DNA technology. First one, treating a host cell with a specific concentration of calcium. Then B, incubating the cell with recombinant DNA on ice, followed by placing them briefly at 42 degrees Celsius heat shock and then putting them back in a ice. Okay, this is what techniques to make a host competent in recombinant DNA technology. Define polymerase chain reaction, name the enzymes used in it. Polymerase chain reaction is a reaction in which multiple copies of a gene or DNA is synthesized in vitro using two sets of primers and the enzyme DNA polymerase. Thermostable DNA polymerase or TAC DNA polymerase is the enzyme used in polymerase chain reaction. Write the method to introduce aligned DNA into host cell. Okay, this is a repeated question, very important question. Recombinant DNA and the bacterial cells are incubated in ice, followed by placing them briefly at 42 degrees Celsius. Okay, and then putting them back in ice by microinjection. The recombinant DNA directly injected into the nucleus of the animal cell. Plant cells are bombarded with high velocity microparticles of gold or tungsten coated with a DNA in a method known as a biolistics or a gene gun. Disarmed pathogen vectors which when allowed infecting the cell transfer the recombinant DNA into host. With an example, explain the convention for a naming restriction endonuclease scientifically. For example, EcoRI, they've taken here endonuclease. Then EcoRI is a restriction enzyme which was isolated from Escherichia coli RY13 strains. The first letter E in the name of the restriction enzyme represents genus of the bacterium Escherichia. Next two letters CO in the name of the restriction enzyme represents species of a bacterium, that's a coli, CO coli. R in the name of the restriction enzyme represents the strain of a bacterium, that is RY13. R represents RY13. Then next, a Roman number following the name indicates the order in which enzymes were isolated. Order in which enzymes were isolated from that strain of a bacteria. Okay. Mention any two methods of introducing aligned DNA into host cell. We already discussed, it's repeated. Okay, I think second time it's repeated. So I am not going to discuss because important points are here. 42 degrees Celsius, putting them in back into ice macroinjection method. They directly introduce to nucleus of the animal cell, then plant cells are bombarded with high velocity. They are coated with tungsten or gold. This method is known as what biolistics or a gene gun. Then disarm pathogen vectors which when allowed infecting the cell transfer the recombinant DNA into the host. Okay, explain the brief separation and isolation of DNA fragments. The cutting of DNA by restriction endonuclease results in the fragments of DNA. These fragments are separated by a technique called gel electrophoresis. Since the DNA fragments are negatively charged, they can be separated by forcing them to move towards anode under an electric field through a medium or matrix. Most commonly used matrix is agarose, a natural polymer extracted from seaweed like Gracilaria or Geladium. DNA fragments separate according to their size through sieving effect provided by the agarose gel. Hence, the smaller the fragment size, farther it moves. The separated fragments are visualized by staining them with ethidium bromide followed by exposure to UV radiation. The separated brands of DNA are cut out from the agarose gel and extracted from the gel piece. This step is called elution. Draw a label diagram of plasmid PBr 322. PBr 322. Okay, here symbolic representation of some words they given. Okay, whenever you are studying briefly this chapter, eco R1, it's a what? E. coli, R1, CLA1, HIND3, PVU1, PST1. Okay, then which one? AMP, TET, ORI, ROP, PVU2. Here PV1 is there, here PV2 is there. Okay, SAL1 is there. These are what code words used in plasmid PBr 322. Okay, this diagram is also very important for 2 marks. Mention the different steps of process of a recombinant DNA technology: isolation of the genetic material from DNA, cutting of DNA and isolation of desired DNA fragments, insertion of recombinant DNA into the host cell or organism, culturing of host cell or obtaining the foreign gene product, downstream processing. Mention 4 tools required to recombinant DNA technology. It's for 2 marks. With reference to gel electrophoresis, what is the elution? Repeated question. It's a tools for required recombinant DNA technology: restriction of enzymes, polymerase enzymes, ligases enzymes, vectors, host organism. The separated bands of a DNA are cut out from the agarose gel and extracted from the gel pieces. This step is known as elution. Write the important use of the following in rDNA technology: PCR, polymerase chain reaction, ethidium bromide bioreactor. PCR, it is what used to synthesize multiple copies of the GNA of interest. Ethidium bromide, it is used to stain separated DNA fragments. Bioreactor, it is used to produce a large quantity of products where large volume of culture can be processed. Then 5 mark questions. Explain the feature of cloning vectors. Okay, features of a cloning vectors are origin of a replication, selectable marker, cloning sites. Okay, these are the what features of a cloning vectors. Then first one, origin of a replication. This is the sequence from where replication starts, and any piece of a DNA when linked to this sequence can be made to replicate in host cell. Selectable marker, it helps in identifying and eliminating non-transformants and permits the growth of transformants. Genes encoding resistance to antibiotics such as ampicillin, tetracycline, chloramfenitol are considered as a selectable markers. Cloning sites, these are the recognition sites needed to link aligned DNA to vector. Ligation of aligned DNA is carried out at restriction site present in one of the 2 antibiotic resistance gene. For example, we can ligate a foreign DNA at a BAM-HI site of the tetracycline resistance gene of PBr 322. Explain the process of denaturation, annealing and extension with respect to polymerase chain reaction, PCR reaction. Okay, define plasmid. A question, B question, and C question. Why restriction enzymes are called molecular scissors? Okay. Then first one, denaturation. First question answer, denaturation. Double strand DNA is denatured by using high temperature at 90 to 98 degrees Celsius for 15 seconds. It results in the separation of two strands. Each strands act as a template for the DNA synthesis. Then annealing. The two set of oligonucleotide primers are used to anneal hybridize at a low temperature of 40 to 42 degrees Celsius along a single strand of a DNA. 10 to 18 nucleotides are formed are called primers. Whatever single strand of a DNA contains 10 to 18 nucleotides are called as a what primers. By using magnesium Mg plus 2 and DNTP deoxynucleotide triphosphate DNTPs. The next extension, the thermostable enzyme TAC or thermus aquaticus. DNA polymerase can tolerate high temperature of the reaction that extends the synthesis of DNA from primers by adding complementary nucleotides to the template. These steps are repeated many times to obtain several copies of DNA, that is 1 billion copies. Then, plasmid is an autonomously replicating circular extra chromosomal DNA. Restriction enzymes cuts the DNA at specific locations. Write the diagrammatic representation of recombinant DNA technology. Write a note on downstream processing. They asked two questions here. Then recombinant DNA technology here. It's a foreign DNA. It's a vector DNA. Okay, vector DNA is called plasmid also. Then same restriction enzyme cutting both foreign DNA and vector DNA in specific point. See here and here. Okay, at a specific point, the enzymes cutting both the foreigns here and here, whatever the cuts occurred. Okay, ligases join the foreign DNA to plasmid. See, this part come here and the whole thing will again come here and they rejoin. Okay, ligases join foreign DNA to plasmid. Then after this transformation, E. coli bacteria is found. E. coli bacteria undergo a cell division. After cell division, whatever the DNA found inside the E. coli bacteria body, that's a recombinant DNA technologies DNA. Downstream processing is the series of processes before it is ready for marketing as a finished product. The process include separation and purification. The product has to be formulated with suitable preservatives. Such a formulation has to undergo through clinical trials as in case of drugs. Okay, up to now we discussed a chapter related to biotechnology. Okay, hopefully this chapter is useful for your exam purpose. ಏನಪ್ಪ ಅಂದರೆ ನಿಮ್ಮ ಎಕ್ಸಾಮ್ ಪರ್ಪಸ್ ಇದು ಹೆಲ್ಪ್ಫುಲ್ ಆಗ್ತದೆ. 1 ಮಾರ್ಕ್ ಕ್ವಶನ್ಸ್ 2 ಮಾರ್ಕ್ ಕ್ವಶನ್ಸು ಹೌದಾ. ಏನಪ್ಪ ಅಂದರೆ ಪಾಸಿಂಗ್ ಪ್ಯಾಕೇಜ್ನ ಪಾರ್ಟ್ ಆಗಿ ನಾವೇನು ಮಾಡ್ತಾಯಿದ್ವಿ ಅಂದರೆ ಈ ಚಾಪ್ಟರ್ನ ಡಿಸ್ಕಸ್ ಮಾಡಿದ್ವಿ. ಹಾಗಾದರೆ ನಿಮಗೆ ಇಷ್ಟ ಅನಿಸಿದ್ರೆ ನಿಮ್ಮ ಫ್ರೆಂಡ್ಸ್ಗೆ ಶೇರ್ ಮಾಡುವಂಥ ಕೆಲಸ ಮಾಡಿರಿ. ಯಾವುದು ಬಯೋಟೆಕ್ನಾಲಜಿ ಪ್ರಿನ್ಸಿಪಲ್ಸ್ ಆ್ಯಂಡ್ ಪ್ರೊಸೆಸಸ್ ಹೌದಾ, ಜೈವಿಕ ತಂತ್ರಜ್ಞಾನ ತತ್ವಗಳು ಮತ್ತು ಸಂಸ್ಕರಣೆ ಅನ್ನುವಂಥ ಚಾಪ್ಟರ್ನ ನಾವು ಡಿಸ್ಕಸ್ ಮಾಡಿದ್ವಿ. ಇನ್ನು ನೆಕ್ಸ್ಟ್ ಒಂದು ಕ್ಲಾಸಲ್ಲಿ ಮತ್ತೊಂದು ಕ್ಲಿಪ್ನ ಡಿಸ್ಕಸ್ ಮಾಡೋಣ ಅಂತ ಹೇಳ್ತಾ ಥ್ಯಾಂಕ್ ಯು ಸೊ ಮಚ್.